I'm Samia Mohinta, and I am a data scientist at the MRC LMB. Our brains are made of cells called neurons. These neurons connect in very complex ways, which determine how we feel, act, and perceive the environment. What happens to our brains when we learn a new skill like playing the guitar, or when we get a disease? Our research is trying to answer these questions by mapping the structures in the brain called connectomes and then linking them to the functions. Studying these neurons in human brains are quite complex because our brains are massive. So we turn to common fruit flies, which have smaller brains, but they are complex enough to teach us how our own brains work. Even with these smaller brains, mapping out these connections in them, which we call the connectome, will take years if done by hand. So we turn to use artificial intelligence, especially computer vision. So we have electron microscopes that take images of these brains, which come out as slices. We stack back up thousands of slices to, again, look like a 3D representation of the brain. What we are trying to do with computer vision is to teach these machines to identify objects in an image. For example, if you go to a zoo and take a picture, a computer vision algorithm can then identify different animals in it. We are trying to teach our machines to do the same thing, but just recognize neurons instead of animals so that we can reconstruct the connectomes faster. A challenge in this field is that these pictures from electron microscope look quite different to what natural photographs look like, so we have to come up with specialized solutions in computer vision to handle this task. People have built foundational models for natural images, but we do not have that for electron microscopy, so we are trying to build one for electron microscopy because we have got so much data to play with. The Accelerate program has given me access to a community of AI researchers in Cambridge who I can contact to bounce off my ideas, and a platform to showcase my research as well. With AI, the models we are currently building on insect brains could be applicable to other species like mice and eventually to humans. On one hand, AI is giving me this unique opportunity to answer these fundamental biological questions. On the other hand, we could take this biological inspiration to build better AI tools. So it's really a symbiotic relationship.